Hello everyone, Paul from Sinobiological. I'm happy to have this chance to give a talk on the topic of the invisible variable uncovering endotoxin risks in recombinant proteins and high standard solutions. I believe endotoxin is so much relevant to you if you are working with recombinant protein, antibody development, or drug discovery. I hope the solutions from Sinobiological can minimize the potential risks as well as your concern regarding the contamination of endotoxins. So my talk will divide it into three parts. Let's go to the first part, the sources and the properties of endotoxins. Firstly, let's briefly look at what endotoxin actually is. So the endotoxin, or LPS, is the major component of gram-negative bacteria outer membrane. It accounts for almost 35% of its composition, and it is estimated that one bacterial cell can contain up to 4 million LPS molecules, and it will release to the surrounding upon bacterial death or lysis. So on the right-hand side, we can see the structure of LPS or endotoxin. So the upper part is the outer oligosaccharide repeating units called O antigen. It is very strain-specific and will define the identity of a bacteria. So the middle part called core oligosaccharide, it is relatively conserved. And the bottom part called lipid A. So the lipid A is the most conserved part in the bacteria and also the biological active component of the endotoxin. So this hydrophobic region adopts a highly ordered structure, also makes it hard to remove. So the next question is how endotoxin matters. Interesting, endotoxin, or more specifically lipid A, does not directly damage cells or organs, but instead it acts as a potent pyrogen, can activate the immune system and cause the people or animal fever, inflammation, or shock. Another thing is like due to their highly stable structure, it is highly heat resistant and can survive from conventional sterilization, which will pose a major safety threat to biologics and medical devices. So now let's take a closer look at the physical properties of endotoxin. The molecular weight of endotoxin monomer is around 10 kilodalton, but normally the endotoxin will self-assemble to a very stable aggregates, and it can form micelles or vesicles, which is even more stable than detergent micelles. So once it forms the aggregates, it's really hard to break them down into monomers, unless you are using very harsh conditions like detergent or Triton or EDTA, which is not very friendly to your recombinant protein or antibodies. It has a very heterogeneity in chemical and physical properties. So next question is why the endotoxin can exist in our surrounding life or even in the experiment. The answer is because endotoxins are ubiquitous everywhere in life. First of all, we can see that humans can be exposed to endotoxin naturally in the environment. We may have endotoxin even in our homes or offices, for example, through contact with dust. It can also exist in water or natural products or even medical devices. In experiments, during purification of any specific molecule like protein purification or DNA extraction, contamination can be introduced in multiple ways, like using buffer prepared with contaminated water or using the endotoxin contaminated materials carriers such as the chromatography resins, or even using the endotoxin contaminated enzymes and consumables will bring the extra endotoxin into your experiment. Actually, the concentration of endotoxin in the biological production process largely depends on the source of the product and the manufacturing process. So ideally, when a product is derived from cell culture supernatant and the whole procedure does not bring any endotoxin in the manufacturing process, the final product should be completely free of endotoxin. However, in reality, achieving a truly endotoxin-free environment is almost impossible. That's because of the endotoxin will accumulated during the production. For example, water quality is one of the critical issues, and even small cumulative amounts of endotoxin introduced through water, it can accumulate a big amount in the process, compromising the purity of the final product. So far, we've discussed where endotoxin comes from and how ubiquitous it is across the different environments or production systems. But more critical questions for us as researchers is, what does this actually mean for our experiment? Does it bother with our data, and how often we potentially mislab without realizing it? So in the next section, I will focus on the impact of the endotoxin in experiment and assays. First, let's see how endotoxin would be involved in experiments. The endotoxin is a potent activator of the innate immune system. It can bind to LBP protein as well as MD2 protein and activate the TLR4 signaling pathway, and ultimately leads to NF-kappa B activation and the production of the pro-inflammatory cytokine release, which cause the inflammation. The LPS can also bind to caspase 4 and 5 to cause the irreproducible and eventually cause the inflammation as well. Endotoxin actually doesn't exist freely. It has a strong tendency to bind to a wide range of biomolecules, including proteins such as albumin. So it's also called a natural superglue. There is a classic case from early study that tried to identify the LPS receptor. And initially, LPS was reported to bind albumin in a 2-to-1 ratio, but this was later recognized as a non-specific binding. And subsequent research suggested that the CD14 or even TLR2 can be the receptor of LPS, but laterly it also incorrect. In reality, the observed binding was due to the endotoxin contamination rather than direct receptor recognition. So finally, TLR4 and MD2 complex was identified as the true receptor of LPS. This example highlights that how easily endotoxin can lead to a misinterpretation in experiments. This is a a strong reason why we are using endotoxin-free recombinant in our experiments. Next, I would reintroduce how endotoxin can have a profound impact on immune responses. One striking example comes from a study on HSP70, which is a heat shock protein known for its immunostimulatory properties. So, early experiments show that HSP70 fused with a tumor antigen OVA can activate the CD8 positive T cells and demonstrate a potential as a tumor vaccine. However, it was later found that much of this immune activation was actually due to the contamination of an endotoxin in the protein samples. And after the endotoxin was removed, HSP70 lost its immunostimulatory effects, demonstrating that the observed immune response was largely driven by the LPS rather than the protein itself. This also highlights that a critical point is even trace amounts of endotoxin in recombinant protein when you are using it for an immunogenicity experiment. The endotoxin contamination can artificially enhance or screw your immune response, leading to a misleading conclusions. Now let's talk about how endotoxin can interfere with antibody generation, particularly when you are used this recombinant protein as immunogens to produce antibody. LPS or endotoxin can non-specifically stimulate B cell proliferation, and this stimulation does not actually make lymphocytes produce specific antibodies. As mentioned in this original paper, a B cell mitogen such as LPS could activate division in B cells, resulting in an increased number of cells producing antibodies to a non-cross-reacting antigens, mimicking the effect of the specific antigen. This is to say that LPS contamination can alter B cell proliferation without truly affecting antigen-specific antibody production, potentially lead to a wrong antibody generation studies. Next, we can also see how endotoxin interfere with the downstream cytokine detections. In this study, researchers try to stimulate the THP1 cell or primary monocytes or other primary human immune cells by trace concentration of endotoxins. As shown in this picture, even very low concentration of LPS can already trigger the release of the cytokines, especially for interleukin-6 and TNF-alpha. Even when the concentration of the endotoxin as low as 2 EU per ml, we can already detect the release of the IL-6 and the TNF-alpha. So this is to say, when you are performing experiments that involve measuring the cytokine production accurately, such as interleukin-6 or TNF-alpha, controlling for endotoxin contamination is absolutely critical. This is to say, even trace level of the endotoxin will trigger or change the release level of the endotoxins, can also lead your experiment to a false data. Now let's take a look at how endotoxin can interfere with cell polymerization and differentiation studies. In This study, it is that the tumor-associated macrophage, or called TAMs, played a crucial role in the progression in the TNBC. So repolarizing their phenotype from M2 to M1 can become an effective therapeutic target. So in this paper, the researcher wanted to induce the repolarization of THP1 derived M2 macrophage towards the M1 phenotype. To this end, the researcher need to generate the pure M1, M2A, and M2D macrophage in vitro by induce them using different reagents. Here, as it shown here, the researchers using endotoxin to induce the M0 to M1 with endotoxin and the interferon gamma. And also they used interleukin-4 and interleukin-13 without endotoxin to induce M0 to M2A. In addition, they also used interleukin-6 and LEAF without endotoxin to induce M0 to M2D macrophage. Here you can see that LPS in this study is a switch for the M1 phenotype. And when you are using LPS as a real functional immune modulator rather than a contaminant, you will know that you need to precisely or properly control the endotoxin for all the reagents you are used in your experiment. You want to add, when you want to add the RPS in your specific experiment, you may need to avoid the endotoxin can be brought from any other reagent. Last but not least, it's also important to know that the endotoxin exposure does not necessarily cause death, but it does make the subject sick, reflecting a strong but non-lethal immune response. So here is an example. There is a protein called TSPO, which is expressed at low level in resting microglia. So when microglia are activated, such as during an immune challenge, the TSPO expression is increased. So here on the left hand shows a result of the measurement of TSPO in the brain using the PET radio tracer. And after the injection of endotoxin into the mouse, you can see the TSPO expression increases when microglia are activated. In addition to the changes in the brain, systematic immune activation is also observed. So following endotoxin exposure, the subject shows elevated levels of cytokine in the blood. And this we will also observe that the subject, like human or mouse, may experience fatigue, headache, muscle pain, or chills. This is to say, like the subject, like the human or mice, do not necessarily die immediately after the exposure of the endotoxin, but they may fall ill. So far we have addressed the risks of endotoxin contamination and how it would interfere or even ruin your experiment. Next, I would like to introduce how cytobiological overcome this hurdle and provide solutions to the researchers. Here we can see that there are multiple standard thresholds for endotoxin level for various biomedical applications. For example, in the industry, the endotoxin limit for water for injection or for single-use consumables is typically 0.25 EU per ml. And also for other highest risk procedures, the limit can be as low as 0.06 EU per ml. In cytobiological, our recombinant protein are controlled to less than 0.5 EU or even 0.05 EU per milligram. This means that even you are working on the most sensitive experiment, the purity or the endotoxin level of our protein is qualified for your experiment. Here, I would say like cytobiological is happy leading the industry standards and provide endotoxin-free solutions to our clients or all the researchers. So if, as we mentioned before, if you have concerns or on the endotoxin risks, including severe animal immune responses after any immunogen injection, or your animal suffered from inflammatory response by unknown reason, or the protein or the antibody or the manufacture process will be interfered by the endotoxin contamination. You may need to try the endotoxin-free solutions. So in cytobiological, we provide pure endotoxin-free solutions, which is surpassed the industry standard and making the protein endotoxin level as low as 0.5 EU per mg or even 0.05 EU per mg. In the meantime, we also guarantee the high purity, high stability, and high consistency, which is ideal for animal immunogenicity studies. So how cytobiological can make the protein so pure in terms of endotoxin level? That's because we are using our pure assurance quality system. From the beginning of the production, we used endotoxin-free plasmids and endotoxin-free buffer with filtration, as well as low endotoxin affinity plastic containers in our Center for Process Center at Houston in Texas. This is a place that does not allow any endotoxin or E. coli contaminations. In the meantime, we also do the regular clean-in-place procedures on site, which can guarantee we have a strict endotoxin monitoring and test procedures. So we have built up a wide range of the different proteins, including antibody drug targets, which is suitable for animal studies, immunogenicity studies, mouse injection, or PK/PD studies. We also have a wide range of the endotoxin-free viral proteins, which is good for immunology or vaccine development. In addition, we also have a wide range of endotoxin-free cytokines, which is good for immunology studies, as well as sensitive cell cultures. Beyond that, we also have some other endotoxin-free immune-related protein covered. Like if we are working with endotoxin-sensitive pathways using TLR protein or APOH protein, we also have the solution. Here, I just showcase two featured proteins from the endotoxin-free targets, which is CD38 and the PDL1. Here you can see we can make the endotoxin level as low as lower than 0.05 EU per mg or even lower than 0.005 EU per mg. In the meantime, we also guarantee the protein has the highest purity and also activity validated. So as I said, we have already built a wide range of the drug targets, which is fall into our endotoxin-free level, which includes the EGFR, HER2, VEGFR, and etc., etc. In this page, I will give you an overview of our cynobiological company. So cynobiological is the global leader in recombinant technology, serving as a premier supplier of high-quality reagents and as well as cell services. We have been working in this area for over 19 years, and our extensive catalog is already cover more than 77,000 reagents. And we also have multiple premium product lines, including the topic in this talk, Prepure endotoxin-free product line, as well as ProViral, the largest viral antigen bank in the world. Those tools are essential for the rigorous drug discovery and the vaccine research. With a track record of over 100,000 successful projects, our core expertise lies in recombinant protection across mammalian, cell-free, and insect platform. We are specifically optimized for speed, scale, and offering a high throughput recombinant protein and antibody production. With a global presence across North America, Europe, and Asia, we dedicate to offer the best solution for you. Now I can hand over to any questions. Thank you.